Hello, thanks for coming out, guys. Yes, I work in Dr. Dattelbaum's lab. I've been there for about 2 years now. I'm going to be talking about two main projects that we do in the lab. And the first is working with luminescent biosensors, which is a great deal of working with proteins and kinetics. And then the second project will be working on sponge ecology and secondary metabolite profiles, which I'll be diving into. I apologize for Dr. D not being here. He started his fall break early, like we all wanted to. And, however, we have fun coming up tonight for research. Yeah. So why do we do research? Anyone? You can just raise your hand, shout out. Why we do research here? You are. Why in general? Anything? Leslie, why do we do research? Huh? Find things out, discovering the world around us and so forth. It makes you feel good, right? Understanding how things work and predicting models and changing certain conditions. And I guess why we do research at the University of Richmond is you can build up your recommendations, you get to know concepts that you go over in lab, in lectures, and you can get to meet students, professors. We're all fun, we're all people like to make bonds, not prevalent or ironic, but personal bond, which is much more stronger. Right, okay. Moving on. The Dattelbaum lab for the proteins work is we work with fluorescent proteins. Now proteins fluoresce on their own. Can you all hear me? This is good volume, right? Great. Proteins fluoresce around 280 nanometers based on the amino acid structure of tyrosine, tryptophan, phenylalanine, and but we can also add fluorescent molecules called fluorophores. And then we can do spectroscopy measurements, which measures the concentrations of proteins in your samples. And basically there are three main aspects of what we do in our lab. The molecular biology side, which is studying the structure of the bacteria, the metabolic processes of your organism. The protein side, which is specifically deals with protein structure and the function in the organism. And then bioinformatics, which deals with the computer analogs and the computer models that can predict and help you out with determining the structure of your protein as well. And this all helps you out with making a biosensor, which biosensors help you detect the amount of ligand in your solution or the change in protein concentration or ligand concentration. So biosensor is a form of detecting. And you want to know how much of some type of ligand in your solution at a given time. So let's dive into it. This is a picture of ribose binding protein. If you all want to take a guess of what it does, it binds to ribose, 5-carbon sugar. Okay. So this structure on the left is our open conformation. First of all, what's a protein? Then just raise your hand. What's a simple definition of a protein?
Inverse rates.
Yes?
Inverse rates.
I hear that. I didn't quite hear that.
Converse rates.
Okay, well, I define a structure of a protein as just a polymer of amino acids, just a straight polymer of amino acids. It can fold into specific conformations, and therefore the folding gives you the desired function. This is one long chain of, and folded into two different domains, which is a hydrophobic core. This molecule right here, this orange bead, is a fluorophore. This is where the molecule fluoresces based on the conditions that we leave it in. In this case, this is the open conformation where the ligand is not bound in this area right here called the active site. Let's see. For ribose binding protein, the fluorophore is is away from the solvent or the outside layer of your protein, and therefore it can fluoresce. So when you measure this, you're going to have a high fluorescence when no ribose is bound. When ribose is bound in the active site right here, the protein goes through a conformational change. And it's kind of like a Pac-Man type thing. You have an open mouth, and then it goes up to those beads and checks, and the conformation changes where this fluorophore is no longer shielded from the outside environment, and the solvent can interact with it. And this leads to a decrease in fluorescence. So how we use this is the more ribose that you have in your solution, the less fluorescence that you have. So this is our graph on fluorescence intensity versus the wavelength. And our fluorophore fluoresces around 480 nanometers. Notice how the black line is our initial condition with no ribose bound. And the more ribose that you have, the less fluorescence you're going to have, because the fluorophore is no longer protected in that hydrophobic core, but is now introduced to the solvent, which will produce some fluorescence. This graph over here is our standard curve, and it's in a sigmoidal shape. The standard curve can help us out with knowing the concentration of ribose. However, we're only interested in this linear path and not so much interested in the ends of it. And the reason being is that the ends, notice here, is 100 micromolar, 1,000, 10,000, and all around the same amount of intensity. So we can't distinguish how much ribose is in a solution if it's above 100, because these all get the same fluorescence measurement. However, we can really get a good concentration value between 1 and about 10 or maybe 30 micromolar or so. So that's a fairly good assay for determining ribose. Okay, practicality's sake, why are we interested in biosensors? Glucose sensing, which is popular for diabetes patients, measures the amount of glucose in your blood for numerous reasons, for regulating the concentration. How these systems work is by pricking your fingers, you get a sample of blood, and an enzyme called glucose oxidase, which converts the glucose into peroxide, and it measures how much is in there, which is it's a safe measurement. It's good, it's accurate. However, it can take a while. Elderly patients often complain about the pain and hassle of pricking your own blood. And our new model system is creating a biosensor which can bind to glucose directly, and that way we can get a real-time assay for glucose concentration without an invasive measurement. All right, so this is the work by Brian Durr, who actually had this published last year, which is a big deal to have something published before you even start grad school. It's a goal of us. But this, with the glucose binding protein, it deals a little bit differently. How with no glucose you have less intensity, because in this case you have two fluorophores that are in proximity initially. When glucose binds to the protein, these two fluorophores separate from each other as the conformation changes and fluorescence increases. So in this case, we have a dissociation curve as like so, and we can measure accurately the amount of glucose in your system between 1 and 10 micromolar or so. So this is also a good assay. The problem with this, I mean, like when I first heard about this, I'm like, oh, great! Like now you can ship it off, and someone can make a good assay, and you can find out what the glucose level is in your blood right off the bat. However, the amount of glucose in your blood is much higher than 10 micromolar. It is much higher, and the amount you cannot measure accurately. I guess my red line would be all the way over here, which would indicate that you can't accurately find out the amount of glucose in your blood yet with this assay. So, however, with the amount of micromolar between 1 and 10, this is a fairly good protein to use. Let's move on. Okay, so now we're going to move on to the second part of what we're doing in the Dr. Dattelbaum lab. This is also in conjunction with Dr. Hill's group and Giles Thompson. I'm working, and this is more of what I'm doing myself. We're working with the sponge Microcyanidum prolifera, which is found in Chesapeake Bay along the docks and shallow shores. And we are growing the sponge at an ambient temperature, which is defined as the thermal maximum that the Chesapeake Bay can sustain for the ecosystems to thrive. And we are growing the sponge at an ambient temperature and at an increase of 2 degrees. And we're just curious about whether or not there are any changes in the amount of secondary metabolites that the sponges produce. And I guess secondary metabolite would be organic molecules that your body uses that aren't necessary for reproduction or glycolysis, like those main forms of metabolism, but are important in your secondary processes. So, let's see. All right, so this graph is an HPLC, which is a high-pressure liquid chromatography. And what it does, if you ever hear the word chromatography, I always remember that it's separation. You want to separate more than one molecule from a bunch of other molecules. So what HPLC does, it takes your sample and runs it and separates it from nonpolar to polar molecules. All right, so let's explain this chart. The blue graph here is the treatment with the increase of 2 degrees, and the red line is just the normal temperature. So our goal was to see if there are any changes in the amount of concentration of carotenoids, which are their secondary metabolites. Now, let me just say that if the red and blue lines overlapped in the amount of intensity, I'd have no project. You'd say, no, there are no changes. Who cares if the water increases by 2 degrees, you know? But however, you can look at these 3 peaks, 1 here, 2, and 3, how there are major differences in the carotenoid concentration. So my goal is to find out what these 3 peaks are, like their structure, and then I work with Dr. Hill's lab to find out how an increase, or in this case right here, a decrease in the amount of these carotenoids will help, or will alter the function of the sponge. All right, so our identification techniques are as follows. The HPLC, we can collect fractions, so we can actually isolate each one of those single peaks, and we can analyze it further. Our mass spec, which analyzes the molecular weight of the compound, but not only that, the certain fragmentation points that can also help us get a readout of what the molecule may be. The Biochemistry, which is a time of flight simulator, it measures the molecular ion peak, so it gives us an extremely accurate molecular weight of our carotenoid, and a UV vis, which gives us another spectrometry tool to help us identify what we have. So this is my HPLC that I ran over the summer, and notice how I labeled my 3 peaks I'm trying to identify. And this is just at the ambient temperature, so the normal temperature, and my goal is to identify these 3 peaks, so let's just dive into it. I first focused on peak 2 because we had a suspicion that it was linked with beta-carotene. These measurements are of UV vis, and notice how this is our beta-carotene, what we think it may be, and this is peak 2, and how this 3-peak shape is fairly similar. I labeled the wavelength because the 2 examples are on different solvents, so they may shift. So I'm just piling evidence to convince scientists that these 2 molecules are in fact the same. This is our mass spec readout. Notice how these specific numbers are peaks that beta-carotene can be labeled as. I read that in some paper that some guy worked his whole life with beta-carotene for some reason, and he said that these specific peaks are beta-carotene peaks. If you have these, you have some type of beta-carotene or sure derivative of this. So my peak 2 is fairly similar in these specific beta-carotene peaks. Oh, this is the Leopold test. Dr. Leopold gave me the idea for it, but it's also called the sprinkle test. In which case, I take my peak 2 this is the HPLC readout, and I sprinkle a little bit of beta-carotene in my sample, and I run it through the HPLC, which separates polar from nonpolar molecules, and I get the same peak with a higher intensity, which means that beta-carotene and my unknown peak 2 carotenoid come off at the same time, which is just more evidence. I'm trying to get you guys to agree with me that that my peak 2 is more like beta-carotene. Let's see the next one. Okay. NMR study. For those who have not taken 206 yet, NMR can help us figure out the structure of many organic molecules through the determination of hydrogen bonds. Let's see. This is my beta-carotene structure. We have this isoprene group, which helps out with the absorbance of light. And I'll be using this. Now, the problem with using NMR right now is that my amount of carotenoid that I have is in very low concentration. So NMR does not work in very low concentrations. However, we ordered a type of tube that actually need maybe 5 to 10 nanograms instead of a milligram of substance. So we're very excited. I'll be doing this fall to figure out whether or not my B2 has the same type of NMR readout as beta-carotene does. All right, so what's next? The BioTOF was actually broken for most of the summer. However, it's now online, it's fixed. I'm actually not allowed to touch it. However, Dr. Dattelbaum and I will be going over this and figuring out whether or not there's 3 peaks. We can get a strong molecular ion weight, so then we can figure out what the structure is.